Hello everyone, and welcome to our lab on virology. So as you've been reading through the lab, hopefully you've come to the point where we are now going to be discussing our glow-in-the-dark pigment and hand washing technique. So hopefully you've at least covered a bit of the other concepts in terms of hair sterility, respiratory sterility, and now we'll be testing how we wash our hands. So I won't be doing the demo here. This is obviously something that's much more effective if you do it on your own. But I want to show you how this works so that we have an idea of it when you come into class. So right now, you see my hands. Don't seem like they have much of anything other than I have a nice popped blister on my right hand. Turn off the lights here, and we see nothing glowing on my hands. I have here a dye that's able to fluoresce under UV light. So this is my shirt under a black light. This is what the dye looks like. What you're going to be doing is you're going to take a little bit of this, dab it onto your hand, and rub it in. What you'll notice is that your hand will show exactly where the dye was spread, and your task is to spread that dye all over your hand and then hand wash it off. Once we're all set with that, we'll meet again to discuss some of the scientific reading. All right, hello everyone. So here's our section on the reading. Now for the reading, you're going to need to find this article at the back end of your lab document, discussing aerosol and surface stability of SARS-CoV-2 compared with SARS-CoV-1. You're going to read through these pages. It's a very short article, 2 pages, so you have this to read here. Look over these figures, and then read this little bit on the last page here, and then we're going to discuss a few things. In terms of the discussion for this lab, y'all will need to access the New England Journal of Medicine Wikipedia page, where you're going to be looking for some of the information that's held on the Wikipedia page, and then also consulting the lab itself. Good luck with that. All right, hello everyone. For our next section of this lab, we're going to be talking about aerosol transmission of the virus. Or any kind of other infectious agents. Basically the idea that the things that live within us should stay within us and not be transmitted outwardly through different things like breathing or speech. So in terms of the SARS-CoV-2, or the virus that causes COVID-19, we begin with the presence of the virus itself. So hopefully these are fairly visible drawings. Yeah, cool. So you'll see a spike protein on top of the coronavirus surface. This obviously has to originate from some sort of organism on planet Earth. We think likely that this originated from either pangolins or bats. Diseases pass between animals to different animals all the time. We just got very unlucky with this particular one. So this virus was held within a respiratory droplet, meaning that as you speak or as you breathe, the liquids that are in your mouth, they contain some of the things that are living or existed in your mouth from before. This is surrounded by water, some saliva, some spit, some other enzymes from yourself, but importantly contained inside of it is live coronavirus, as of course if you are infected with the disease. Now this respiratory droplet needs to stay wet in order for the virus to stay alive. As long as it's wet and alive, maybe it's circulating through the air for short bits of time, as y'all hopefully remember from your reading, or it could travel directly into a person's eyes or mouth or nose. Now, once that travels from inside of me through the air into another person, we then can follow the path of infection inside that particular individual. So once the coronavirus gets into a new person, it will enter through those same liquid surfaces. It needed to be in a droplet to be stable. It needs to get into you via your mucous membranes, either your mouth, your nose, or your eyes. If coronavirus were to, say, land on your skin, my skin is its own membrane. It's not allowing anything in. It's impermeable. It's its own barrier. I have oils. I have different substances on my skin to help break different things down. So it's really when this respiratory droplet carrying coronavirus actually gets into some of your wet membranes as a person. Now, once it's inside, you will then have this virus find its way to a receptor called the ACE2 receptor. Now, this enzyme, or this receptor rather, is present in all of us. It's one of these necessary things that we have in order to regulate our heart rate, actually. But it's also the door that coronavirus uses to get into us. The coronavirus has a receptor binding domain, a particular region of the spike protein that's going to bind to the ACE2 protein on our cells and will then get in. Once it gets inside, it is then able to steal your cellular machinery. It bumps into ribosomes, which are your protein factories. Those ribosomes are going to read the mRNA from SARS-CoV-2 and then they're going to make a whole bunch more of that infection. It's going to keep replicating up until that cell is going to explode, pop, and spread around to other cells. Kind of wild, right? And that's the whole name of the game here. It lives in a respiratory droplet from an infected individual. If that respiratory droplet were to dry out, the virus decays. It does not have a good ability to stay alive without being in water. So that's a good defense mechanism. But once it's transmitted to the eyes, mouth, or nose of an uninfected person, it will then bind to the ACE2 receptor, get inside the individual, co-opt the ribosomes, co-opt the entire cell, and begin to proliferate. So hopefully this makes sense as to why someone who's wearing proper protective equipment, like a nurse or a doctor in a hospital who's got their mask, their face shield on, why they can safely deal with patients who are positive for coronavirus, or hopefully more safely deal with them, than someone who's not wearing proper PPE. As a demonstration, I don't want to use acronyms without explaining them, PPE, personal protective equipment. I'll be throwing that around a fair bit more. As a way to demonstrate just how effective some of this PPE is, I want to show you my ability to blow out a flame, blow that out pretty easily on my own. If I am wearing, say, a cloth mask, so I've brought in a old t-shirt to demonstrate approximately the thickness of cloth that you get in something like a bandana. So I will cover my mouth up here. Easy peasy, right? No issue in blowing out a candle or a lighter here when I have just cloth on. So what happens when I'm wearing an N95? Now keep in mind this is an N95 that I've worn for a couple weeks now, but let's see how effective it is. Put it on. Snug against my face. I've pressed there. I have no barriers here between my chin. It's all compressed. Nothing, right? The only thing it's getting is maybe a little bit of backlash, and that's me blowing super hard. So hopefully that demonstrates to you just how effective these masks are at preventing respiratory droplets from traveling a distance. As it turns out, masks really do work. So one of the other pieces that we want to chat about in terms of aerosol transmission, because I've heard this used a lot in different groups discussing things. What's the difference between airborne and droplet? Because the masks are incredibly good at preventing droplet transmission. They're pretty good at airborne transmission, but droplet transmission in particular. They're thick enough that they're actually catching a lot of that water. I'm not talking about spit that you see coming out of your mouth. I'm talking about the microscopic water droplets that are simply just coming out as you speak. You're not going to see them. The mask will certainly still be able to catch them as long as it's filtering well enough. But what about this difference between a virus that's truly airborne, meaning that it does not need that water droplet, versus a virus that needs that water droplet, like coronavirus? Well, airborne transmission occurs when a virus is, as I mentioned, able to survive without water around it. That means it's shelf-stable in any environment, whether it's surrounded by or not surrounded by water. These type of viruses are horrendously transmissible. For example, maybe you've heard of the disease measles. Measles is one of the most infectious agents known to man. If you have a room full of people, 90% of the people who are not vaccinated or not protected against measles, if there's one individual in that room who has it, 90% of the non-protective people will walk out of that room with it. That disease is able to survive on surfaces, it's able to survive in the air, it's able to circulate through the air. It does not need to be encased in a respiratory droplet, meaning that drying out does not end its life. And because of that, measles is in our history books, hopefully, as one of the most contagious diseases ever known to man. Contrast that with diseases that are transmitted via respiratory droplet. If you're via respiratory droplet, a mask will catch a lot of you. Simply time in settling will catch a lot of you, and that time to decay will also be very helpful, right? We're able to use Mother Nature as a means through time just to kill this virus, rather than being worried about the fact that we are spreading this continuously through our air vents. That is a huge benefit to us. So hopefully that gets some of the brain juices going as to thinking about how this virus travels between people, how it gets inside of your cells, how it replicates, how it steals that machinery, connect that to some of the different things that you're learning in this class, and answer the questions attached to this part of the video segment. After you're done with that, I'll see you for our discussion of our internal defenses. Because keep in mind, whether it's the mask, whether it's gloves, or whether it's proper clothing, all of those are preventing the disease from getting inside of you, right? They're preventing entry in the first place. We're now going to switch to talking about the things that protect you once the disease is inside of you. I always like to say the mask protects you from getting the disease in the first place. The vaccine protects you once the disease is inside of you from wreaking havoc. So you have both. People with a vaccine, I'm sure you heard, people with a vaccine can still catch COVID. But their symptoms are much lighter, sometimes asymptomatic, not requiring hospitalization. Mask if you don't want it at all. Vaccine if you want to make sure that you know what you're doing once it's inside of you. Have fun answering those questions. I'll see y'all at my desk when we discuss our internal defenses. All right. Hello, everyone. So we're going to now discuss our internal defenses and what's happening inside of us rather than what's happening outside of us as to how COVID transmits. So let's take a look at a couple of drawings first. So connecting with what we've chatted before, we begin with a live coronavirus. You can see the spike proteins here with the red triangles on them. The purple indicates the inside of the cell, and then we of course have our mRNA sequence in black. That is the stuff that is actually the protein coding regions of the coronavirus genome. Now, the live coronavirus will be introduced to a person who is uninfected. It will go into those via respiratory droplet. It'll go into those mucous membranes of that individual and infect their cells. Now, once it gets into a cell, you'll see that it's able to enter via the ACE2 receptor. Here you have all the different components of the cell that we've talked about in this class. And what's happening on a molecular scale there is that the mRNA genome of the coronavirus is being copied into proteins via the ribosome, and you're making more and more copies of different coronaviruses, occasionally having their own mutations along the way. Now, when this particular cell has made enough of them, it will pop open, it'll burst, and start infecting other cells as well. In this way, SARS-CoV-2 is able to get into any cell with an ACE2 receptor. So, what kind of cells have ACE2 receptors? Well, these are cells that need to regulate blood pressure. So anything associated with your circulatory system is going to have an ACE2 receptor. This means that it's able to get into cells in the lungs and explode them. And if enough of them explode, you'll end up on a ventilator. It's able to get into cells along your bloodstream and pop them. It's able to get into different components of your kidneys or lungs, or even, in some folks, their brains. So it's able to get into a lot of different cells and cause damage to those tissues. Now, when an individual is infected with SARS-CoV-2, they're said to be shedding the virus. Viral shedding is simply the fact that your body keeps making more and more of these unwittingly, because again, the virus has co-opted your system, and in making these, they're ending up in your own respiratory droplets in order to spread to other individuals. Some of the places where they'll hang out in those respiratory droplets are like in your nasal cavities. This is why when we perform PCR testing for SARS-CoV-2, for the coronavirus, we're going to swab your nose. We're going to see whether there are mRNA sequences from SARS-CoV-2 present in your nasal cavity. So that's why testing is done back in the nose, so that way you can actually see whether there's a reservoir of the virus hanging out. Now, once it's in your system and it's exploding cells, your cells are going to have a lot of different methods to communicate the fact that they've been co-opted. There is a red panic button in these cells that say, hey, immune system, come check me out. I'm in trouble. Something's taken over my ribosomes, and you should probably destroy me. That's right. When a cell has been infected and it recognizes that it's been infected, it's going to recruit the immune system to destroy that cell. Your immune system eats your dead or infected cells. But how does it know to do all of that? How does it know which cells are infected and which cells aren't? Well, we'll go over that a little bit later in the class, or when we've discussed your immune system. But for now, know that there is that button that can be pressed, and we're going to instead focus on how your immune system can respond more quickly to an infection, particularly if it's seen that infection before. And this, of course, leads us to our discussion of vaccines and the horrible history of diseases in humans. So here on the left, we have a child infected with measles, that incredibly contagious disease that I mentioned earlier. We have a former president of the United States infected with polio, causing him to lose the ability to move his legs. One of the few pictures of FDR with that, because he did not want to be seen this way. And then we have another horrible disease, smallpox, that's only been eradicated of late. So let's talk real briefly, specifically about this horrible disease of smallpox, causing fevers, vomiting, blisters, blindness. It's spread by close contact and fluids. It creates a lot of pustule packets on your skin. Really gross. It's caused by the virus variola virus. It's one of many different variola genera, and it's been eradicated in the wild, with the last case occurring in 1977. So certainly within the lifespan of your parents. Now, the way in which we learn how to fight smallpox, because you can get the disease and you might learn how to fight it. You might learn before you contract blindness or there's too severe damage. These marks, you best believe, will leave scars. So again, you can learn how to fight the disease by actually getting the disease, as long as you don't die. But fortunately, folks have been working on the idea of vaccines for quite a long time. And this story dates back about 200 years to around the time of our founding fathers as a country, America, where a scientist in Britain noticed that one of his milkmaids, Sarah Nelms, became infected with cowpox. And these milkmaids, generally speaking across Britain, would not have a severe reaction to smallpox as long as they had gotten cowpox before. So what this individual did was he took a child of one of his laborers on the property, James Phipps, and inoculated this kid with cowpox pus from Sarah Nelms, who had at the time a mild case of cowpox. Now, when he gave James Phipps the cowpox, he also fell ill with a mild case of cowpox. So a disease that went from an animal to a human, and then from a human to a human. Now, then, this individual took scabs from an individual who actually had smallpox, the much more virulent, much more disastrous strain of the disease that infects humans, and in a wild case of ethics, decided to then expose Phipps, the small boy, with smallpox. Now, Phipps did develop smallpox, but though he was exposed to smallpox, he did not have a strong reaction to the disease. They noticed that the previous exposure to cowpox gave you protection against smallpox. The idea here being that different or weaker strains can give the individual the ability to identify the variola virus family and eliminate the infection before this happens. So, you get a mild case of something to learn how to fight the thing before it gets severe. And this is the basic principle behind vaccines. We give you things that will teach your immune system how to identify a future combatant. In the case of smallpox, the first versions of the vaccine were literally just the disease from a different organism. Cows are different from humans. You can take a strain that's designed to be really bad in cows that does not infect humans super badly and teach our immune systems how to work to fight off future infections of smallpox. A similar idea has been worked out for measles, but there wasn't a case where we can get a weaker or attenuated strain of the virus from a different organism because measles is specific to us. Cool. Thanks, nature. So measles is a disease that causes a fever of up to 104 Fahrenheit, rash, diarrhea. It's extremely infectious. Viral and airborne, as we talked about earlier, and it's endemic, meaning that it will continuously be present in human populations. We cannot totally eradicate it, but we can protect you from it with a vaccine. So the vaccine for measles is an attenuated form of the virus. So this means that we weaken the virus by having it grow in another organism. In many cases, these are things like chicken embryos, where the virus will replicate in something that isn't human, get really good at dealing with that organism, and then we reintroduce it to us and go, hey, guess what? You got good at dealing with that, meaning that you're worse at dealing with us, and now we're cool. Now we can identify that disease before it wreaks havoc and does this to small children. Now, the vaccine for measles, because this is something that unfortunately has cropped up its ugly head once again. The vaccine for measles is not correlated with Guillain-Barré, the autoimmune disease where you attack yourself. It's not correlated with autism. There is no correlation between vaccines and autism, as much as the pseudoscience might be pushing that. The paper that discussed this, I'm sure I will have discussed this in class with you, but the paper that first published that link was under heavy scrutiny, and the author of that article literally came clean and said they made up the data and retracted their paper. So, no scientific evidence between the tie between autism and vaccines. Nor is it associated with irritable bowel, or inflammatory bowel syndrome. Most side effects of the vaccine are short-lived, a short fever, a short rash, in contrast to getting the disease which may leave you with permanent damage. So I brought all these horrible images to hopefully remind you to not forget how different the human experience was before vaccines. We are incredibly lucky to live in a world where so many horrible diseases are no longer a daily threat to us. The idea behind vaccines is that your body recognizes foreign things and eliminates them, but it takes time to develop a good recognition pattern. As mentioned during the smallpox section, you can get the disease and you can learn to fight it off. But in the meantime of you learning, it is ruining your body. A vaccine does the learning without the ruining. It trains your immune system to have a good recognition pattern without the virus actually damaging your body. Early recognition prevents large-scale spread and damage in the human body. So what about the COVID-19 vaccine? Because we've heard that it's slightly different. It's an mRNA-based vaccine, right? We're not giving you an attenuated form of the virus. We're sticking nucleotides inside of you. Scary? No. I can't even fake a scary voice with this. The idea with the COVID-19 vaccine is that rather than first being exposed to the virus, getting very sick, and learning how to fight it off, we instead are going to give you that search pattern. The mRNA sequences to make spike proteins. They get inside of you. You learn the search pattern, and sometime later, you may be exposed to the virus. Now, once exposed to the virus, rather than getting potentially very sick, folks who are vaccinated may have minor symptoms. And so on a cellular level, what this looks like, the development of the vaccine in the first place and all of the extensive human trials that were done to make sure that this is safe, is we started by sequencing the SARS-CoV-2 spike protein, the genome, and the entire virus. We then synthesized an mRNA molecule and put it in a fat globule. A fat globule, if you happen to be receiving the Moderna or Pfizer vaccines. Pfizer's it needs to be kept at minus 80 degrees Celsius before going into you, whereas Moderna's a little bit more shelf stable, just needs a refrigerator, but it's a fat globule nonetheless. Johnson & Johnson's vaccine, instead of a fat globule, uses an adenovirus, something else, a protein-based mechanism of delivery, but all of them fundamentally just rely on this spike protein mRNA. We then put that mRNA inside of you. It gets into your cells in the same way that coronavirus would. So I always like to point out, if you're not scared of the coronavirus, the vaccine is acting the exact same way, it's the exact same materials, without the damaging side effects. So I'm much more in favor of the vaccine than I am of actually getting corona. Now, once it's inside of you, you will then produce those spike proteins, and rather than exploding that cell will stay alive, stick those proteins on its surface, go, hey, immune system, come on over. Let's launch an immune response. Let's go ahead and remember how to destroy this quicker the next time that we encounter it. And in this way, this vaccine does something different than our other vaccines. It's actually giving us the material to make the spike proteins, giving us the mRNAs to make the spike proteins, and launching our immune response intracellularly. With previous vaccines, our science has only developed to the point where maybe we were giving you a whole bunch of this protein. Or maybe we were giving you an attenuated version of the live virus. There's no live virus in this mRNA. It is just one small protein from it, so a lot less risk. Polio was one of the diseases that did have a live virus, and as one blemish to the history of vaccines, there was one case in Africa where a batch of polio had mutated and was therefore infecting people with a live strain of polio. But that's with live viruses. None of the COVID vaccines are live viruses. Very important to understand this. And so, in this way, we are hopefully able to protect you from the coronavirus by giving you and your immune system a search pattern to recognize and destroy the virus before it gets inside of you. Or rather, once it gets inside of you, before it causes a lot of damage. So, with that understanding of how vaccines work, I now want to take a moment to discuss, hopefully, something that will not be an issue, but something that is obviously present in the popular discussion now. Variants of the virus. Variants of SARS-CoV-2, particularly the Delta variant. Y'all have seen it. That's your Greek symbol written out. Nothing to do with Delta Airlines, as their CEO is quite pissed off about it being called Delta, but c'est la vie. This variant of the virus we understand to be far more transmissible, meaning that it's easier to go from person to person, but not that it changes the pathology or the symptoms of the disease that much. It seems to basically do a lot of the same things, respiratory illness primarily, but its ability to spread is much, much, much stronger than some of our original strains of coronavirus. This kind of evolution is normal. Viruses mutate all the time. The more people get infected, the more opportunity they have to mutate. Since a lot of people have been infected with SARS-CoV-2, it had a lot of opportunity to mutate. In comparison, however, SARS-CoV-2 mutates extremely slowly compared to, say, your influenza virus or your rhinovirus that's causing your common cold. Your common cold will mutate 50% of its genome over the course of a single flu season. So 6 to 8 months, you've got 50% difference in all those proteins and the spike proteins, meaning that your flu vaccine this year might work for this year, might, but by next year, it's a totally different strain. 50% of it is totally different. If your friends changed 50% of their face, I bet you wouldn't recognize them. And that's the same principle with the immune system. So what about the Delta virus, the Delta variant of the coronavirus? Well, let's look at some of the statistics. The entire SARS-CoV-2 genome is approximately 30,000 base pairs big. If you want an exact number, it's 29,870 nucleotides long. This is its entire genome. The S protein is just one part of it. The S protein is approximately 13% of that entire genome. It is exactly 1,273 amino acids long, meaning that it's 3 times that base pairs. So that's where I got the math for this, right? Not super important, but gives you an idea of how big these things are in terms of some of your foundational molecular building blocks. Now, how does the delta variant's spike protein, and here's what the spike protein looks like. You have nucleotide N-terminus domain. You have a receptor binding domain. This is super important because this is your region that's going to bind your ACE2. And then you have some other structural components of the S protein. This is actually what our biochemistry model of the S protein looks like. It's got two different sections, subunit 1 and subunit 2. Here's like a structural domain with a whole bunch of helixes. You got a thing off to the side here. And then you've got this blue region right there being your receptor binding domain. Now, the receptor binding domain is typically tucked in, but when it bumps into the right chemistry, that receptor will swing up and out. As it swings up and out, it will bind to your ACE2, and then you can get the spike protein helping you get into the cell from there. So, what's different between the Delta variant spike protein and the original SARS-CoV-2 when it first entered human populations, or was first sequenced at least, in January of 2020? Well, when we compare it to the original strain, we see that the spike protein has 8 mutations and 1 deletion, meaning that there are 8 different amino acids, or the foundational building blocks of a protein, and 1 deletion, so 1 less amino acid. Now, 4 of those mutations in the Delta variant occur here. There are 4 in the N-terminus domain, meaning the front end of it. And then there are 2 in the receptor binding domain. They get their own names. Wild things like L452R. Now, the way this notation usually works is this indicates one amino acid, this indicates the location, and this indicates the change to what it is. So the other mutation in the receptor binding domain is T478K, meaning that you change a T to a K at amino acid 478. And you see how the receptor binding domain goes from 319 to 541, meaning that both of these occur in this region. Kind of crazy how at first something like L452R means nothing to you, and then all of a sudden it means a little bit more than nothing, right? I know that the script here is kind of opaque, but hopefully you're able to follow along. And then we have some other mutations down here in the other regions, the protein. Whoop. So I mentioned this mutation in the receptor binding domain because that's actually what some folks think causes delta to be so different. They think it actually reduces the specificity of binding to ACE2, meaning that it doesn't need as tight of a lock in order to get into our cells. Some of the other mutations, we can actually see all of the mutations associated with the Delta strain right here. There's a website that tracks all of this, lots of information online publicly available about this. But you can go and find all of the mutations present in the original Delta strain and subsequent Delta strains in different states, in different countries. And you can see all of them. They all have their names. They all have their identifiers. You'll notice that, for whatever reason, this open reading frame 1 up ahead doesn't seem to have any mutations. We've got an open reading frame here in Delta that has just a couple. A large concentration in the S protein, that incredibly important protein that is what we've designed our vaccine around. And then some mutations further down in other proteins. But in terms of the efficacy of the vaccine, remember that the S protein was 1273 amino acids long, and we've changed 8 of those 1273. meaning that less than 1% of the amino acids of the spike protein, approximately 0.6% of those amino acids, are changed between the Delta variant and the variant that your vaccine was designed for. Is a 0.6% change huge? Maybe in the viral world, but it actually turns out that it's not. This is why now what we're hearing discussed, and this, for full disclosure here, this lab is being recorded in fall of 2020. The exact date is August 23rd. So information is current as of then. But with the Delta variant having a very small difference rate in the S protein, we're seeing that the vaccine that we've designed is actually effective against Delta variants. Delta seems to spread more easily, but the vaccine still protects people who have been vaccinated against it because there is just a smaller amount of change, or such a small amount of change, that your immune system is still able to recognize whether it's a Delta S or the original SARS-CoV-2 S. And this will remain true as long as strains do not mutate a huge percentage away from the original mRNA vaccine. So why a booster, right? Because there are two ideas behind why you need a booster. Well, if the S protein changes too much, you may need a booster in order to be able to identify what that new S protein looks like. If, for example, 50% of it was different, you'd best believe that there will be a booster that teaches your body what the new S protein looks like. But that's not the case here. Instead, we're looking at booster shots to help ramp up antibody production. I apologize for my terrible handwriting. Goodness gracious. Antibody production. Because your antibodies are not inside of your cells. They're floating around everywhere. Imagine that you had an individual break into your house at midnight. You heard them, maybe your dog alerted you to their presence. You might be on high alert for the next 6 months, right? Like any kind of creaking cabinet, any house adjusting, any tapping on a window, that's going to wake you up much easier, right? You will be on high alert. But maybe over time, that high alert status, if there isn't another break-in, you won't be on such high alert. Your immune system acts in the same way to a large extent. That high alertness is actually embodied by your body making a whole bunch of antibodies, these free-floating proteins that are sticking to SARS-CoV-2 to prevent it from binding to any of your cells. But this ramped up antibody response is only present for something like 8 months, meaning that your body detected the presence of the virus when the vaccine was present inside of you. It designed a whole bunch of antibodies to bind the S protein, and now those antibodies are going to stick to the S protein for the next 6 to 8-ish months if you encounter SARS-CoV-2 again. But your body goes on lower alert if it's been a while since the vaccine or since the vaccine. So therefore, these boosters are to help ramp up that antibody production to hopefully eliminate SARS-CoV-2 from the population in the first place. We're trying to wipe this thing out. There's no need for it to continuously circulate across human populations. If we can get everybody to be vaccinated and everybody to have high antibody count in them, we're going to reduce the severity of the disease in people who do contract it, and then also reduce the spread of the disease in our populations. Now, hopefully a lot of this has made sense to you. hopefully a lot of this vaccine discussion and the presence of mRNAs has been informative. But because of recent headlines, I'll end on discussing one other thing that is sadly comical. A compound called, or drug called, Invectrim. Invectrim is something that helps horses deal with parasites. Somehow through social media, some folks have been convinced that this is the actual solution to treating your SARS-CoV-2 symptoms when you get COVID-19 from the virus, rather than getting the vaccine. Keep in mind, hopefully I've explained to you everything that's in the vaccine, fat bubble with mRNA, so that way you can learn how to make the spike protein, or adenovirus with mRNA, so that way you can learn to make the spike protein, all compounds that get broken down very quickly, that interact with your immune system, that teach you how to fight. Contrast that with Invectrim, something that's designed for horses, against parasites, that has had no clinical trials to show any efficacy in combating COVID-19. And yet, there are folks ingesting this when they catch COVID-19. And they're instead ending up in the hospital, potentially for both, but most certainly for Invectrim, because this is not going to be something that gives you reasonable side effects, nor will it do anything to battle your COVID-19. So it boggles my mind that people are willing to listen to somebody on the radio or a tweet and take something that has no doctor recommendation, contrast it against vaccines that are highly efficacious, that do help protect against severe infection. And hopefully now you understand how they protect against severe infection rather than protect you from getting the disease in the first place. Mask up if you don't want to get it in the first place. Vaccine up if you want to know how to fight it quickly and prevent severe side effects. So with that, I will end this video discussion and leave you to discuss the questions at the end of the lab. Cheers. Back at you. I know the last video said that we were done, but it's a few days after I finished recording that, and now it's after the welcome event where I spent some time building a Lego model of the S protein. It's kind of fun to put together. I wanted to touch bases to a couple things and something interesting that happened at the welcome event for West Shore here when I was building this Lego model to try to convince you that actually building things and playing with them can be an educational experience. So here we have the paper from which y'all will recognize. It's a cool paper. It talks about the native state receptor binding, intermediate, how it forms the connection, and then how it actually gets through, cell fusion, how it gets in, and then how it lets go. So we have all this modeled, and then y'all will recognize this picture, right? This is the piece that we were talking about before. So I want to model what this looks like. So down here, you'll notice that here are all these alpha helixes, these repeated regions of very particular amino acid biochemistry. Whoa, big words. Really structural pieces of the protein. They're at the base. You've got a clip here off to the side associated with the subunit 1 of the S protein, and you've got another larger piece off to the side here for the oh, sorry, that was inverted. So this large piece here that actually extends up, you've got one alpha helix off to the side, and then you've got another alpha helix here that has this other structural region that binds to the receptor binding domain. So you have the open state of it where it's flopped up, and then you have the bent down shape of it. And so now that hopefully we understand that, I'm going to enlarge the screen here on myself so that way we can see what cool insights were gained from this. So the protein model in its full Lego glory. And then we, of course, have the ACE2 protein. Now, the lock and key model is something that we will have or have talked about in this class, where proteins will fit together quite perfectly. So you'll notice that in my first iteration of this, I built the receptor binding domain with a very particular pattern and the ACE2 protein with a complementary pattern to that. Now, just playing with this, I was like, okay, well, so how easy is this going to be to fit together? So then I take the ACE2 protein and simulate binding. So then I fidget around with it a little bit, and it fits. It fits perfectly, just like you would expect with Legos, and it's able to bind. So then I wanted to say, well, how can I model this being different with Delta, right? Like Delta is pretty much exactly the same thing, just with a couple mutations. Maybe one particular one in the receptor binding domain that causes it to be less specific. And so if I wanted to model less specificity in this receptor binding domain, I'd just take off a piece of Lego. And now they don't fit perfectly like a lock and key, but they can still fit together, right? They still have enough there. So this piece is now less specific. And then I took this piece, the ACE2 protein, and stuck it on. And it happens even easier. Now that's fairly counterintuitive, right? When reading the paper, I didn't really understand this, and maybe I'm entirely making this up, also a possibility. But with less specificity, it's able to form this connection, this weaker connection, this starting connection, perhaps quite readily. And then importantly, the piece that's actually doing the lifting for actually getting this into the cell, so if we look back at our image here, the receptor binding is first, but then you have this intermediate step. This intermediate step facilitated by this piece here, right? So when we bring this down and connect the pieces, this piece here is now close enough to the ACE2 protein to actually form a very strong bond, and it's facilitating this cell fusion. And so in my own head here, and I ran this by the folks from the health department, and they thought it sounded believable at the very least, and ran it by a couple of my friends who know a fair bit more about protein biochemistry than I do. It sounded believable as well, is that maybe this receptor binding here, this mutation allows for like quicker reactions, but the strength here is actually still allowing COVID to function essentially the same. Again, no scientific evidence of this. I'm hand-waving here, but it's at least cool that it's plausible. And to make sure that I'm not crazy, right? I'm playing with Legos, and I got this in my own head. Maybe it's confirmation bias. I brought this model out to the welcome event yesterday, and had a large number of people try to mess with the Legos and get it to stick on top and see whether it was easier with or without the Delta mutation. And everyone said it was easier when it was less specific. That's super counterintuitive to me. I'm still wrapping my head around it. But I will have this Lego model in the classroom, and I'm very surprised as to how much benefit there was of playing with Legos and building protein models. That was not something I expected. I thought I'd maybe have a nice, pleasant day in the sunshine building some Lego models, and I think I learned something kind of cool, or at least got more excited about protein biochemistry to read some more about that. So if you think that you might want to play with some Legos, maybe justify the purchase of some Lego kits for the classroom, please do let me know. This is actually going to be the end of the lab. I promise not to record another stint in the lab, but I do encourage you all to play with the Lego model and hopefully learn as much as I did, or at the very least get more excited to continue to read more, because COVID's not done evolving yet, for worse or for worse. And so we're going to have to track these things. We're going to have to see what's going on. But it really does elucidate how much we do know about this virus, how much we understand, how much we don't know too, right? Like I'm still hand-waving on this. But the fact that we're able to map this out in such a precise way actually really makes me feel much more confident in the science around all of this, right? So with that, I will actually end. Have fun playing with some Legos, and I do look forward to working with y'all throughout the rest of the semester.